All right. Hi, guys. My name is Natalie, and the pathogen that I have decided to research for this project is Mycobacterium leprosy. And this photo here on the left is an acid fast stain. And as we've learned in the class, we know that the mycobacterium cells are going to be the red cells. So I want to begin with this case study. So some researchers found, I believe this was in 2016, they're still kind of researching it, that leprosy was found in chimpanzees for the first time ever. So, some wild chimpanzees in West Africa, there were two in different national parks. They were found to have contracted the Mycobacterium leprosy for the first time ever. And it's been known that leprosy can be contracted and found in armadillos in America and then red squirrels in the UK. And they share the same bacterial genotype of 3I, which is a genotype that has been found in the ancient outbreaks that have happened from medieval Europe and basically the same genotype that we've known of for this disease. However, the genomes found in this leprosy in these two chimps are known to be rare, and they're known as 2F and 4NO. So this specific case, it ruled out human transmission because it would have required close contact, and there were no cases in this study that they had humans in contact with these chimpanzees. And researchers believe that because of this, there may be an unidentified leprosy reservoir, or possibly that the bacteria had another host before human evolution. So, our intro into M. leprosy, it's most commonly known as Hansen's disease, based off the person who discovered leprosy, Hansen, or it's just known as leprosy disease. And it comes from the Mycobacterium genus. It's very slow growing. So, it has an incubation period of about 1 to 20 years, 5 years average, but it's often been prejudiced and feared because of how contagious it was or how it thought it was contagious back in the day. We'll see further in this presentation that it's not as bad. And the trauma that was basically just linked with it. And it's been around for many centuries. It's often implied in biblical literature and ancient civilizations. And those who contracted this disease in the time period were often looked down upon and excluded. But we see that research over the years and decades, it's really helped isolate this disease. And in 1 year, there's really only 150 people who get it in America and 250,000 globally. Now, this is a super small number compared to your typical bacterial infections. And then we'll go on with our characteristics and morphology of this pathogen. So, Mycobacterium leprosy is an acid fast bacteria. It is rod shaped or a bacillus. It's 1 to 8 micrometers long and zero point 0.2 to zero point 0.5 micrometers in diameter. So, as we've learned with Mycobacterium, they contain a fatty acid called mycolic acid in their cell wall. It's very lipid rich, waxy structure, and because of this, the primary stain in the acid-fast staining technique is carbonyl fusion. So most stains, they can't penetrate this lipid cell wall. So carbonyl fusion is more lipid soluble and it's able to bind to the mycolic acid. And then methylene blue is going to be our counter stain, which will stain the non-mycolic acid cells. So if we see on these two images on the right here, both of these are acid-fast stains. So we have clumps of acid-fast cells in a macrophage. And then on the bottom, we have leprosy in a lung of a 27-year-old male. So the mycolic cells are going to be the red ones. And then this picture up here just shows kind of the shape. You can see that rod-shaped cell of the Mycobacterium. And leprosy, it has the longest doubling time of 13 days. That's the longest doubling time of all bacteria. And isolating and incubating this can be super difficult. So, how researchers research this bacteria is through the footpad of mice and armadillos to examine the cells. And it's also examined on human skin cells as well that have been affected by it. And it thrives slightly below at 37 degrees Celsius, which is why it loves the skin, and it also can affect nervous tissue when absorbed into the blood. So here is a picture of the cell membranes of some common bacteria. So this is a gram-negative bacteria, a gram-positive bacteria, and then right here are mycobacteria. So we know that gram-positive has a lot of a more of a thicker cell wall because it has a thicker peptidoglycan, as shown in the purple. But for Mycobacterium, it has these three main components. So the first one is that cross-linked polymer of peptidoglycan, and then we have something called a arabinogalactan polysaccharide. This right here in the blue. This is a component that gram-negative and gram-positive cells do not have. And then lastly, most of all, we have the mycolic acid here in green, the long chains of mycolic acid. And it gets really difficult for a common stain like crystal violet to penetrate through a cell like this, which is why we have to have carbonyl fusion. And then for our causes and diseases of this illness, it's found in armadillos, actually. So in the southern region of the US, a lot of armadillos, they naturally carry the Mycobacterium leprosy. So it is known as an animal reservoir, but it is able to live and thrive in humans. However, transmission from animal to human is often really rare because you have to be in long periods of close contact to get it transmitted. The most common transmission of this is from coughs and sneezes. Now, even though this disease has been around for a really long time, scientists still have not found the exact source of transmission. But they have found that it's not as contagious as it seems. There's a common misconception that skin to skin contact will cause a spread of illness. And we'll see later that there are some photos of what it can do to the skin, and that's why people thought if they were to touch someone with leprosy, they would get it as well. And another misconception, it's actually not transmitted through short spurts of close contact, like hugging, sitting together, etc. And then here are our signs and symptoms. Here are those photos I was talking about. So it most commonly affects the skin. It's like a visual symptom. So it can result in discolored skin patches, nodules, thick, dry skin, foot ulcers, swelling and lumps, and even loss of facial hair. So this photo right over here, we see those discolored skin patches, some bumps on the skin, but also can affect the nerves. So the bacteria can get into your blood vessels, and that can result into nerve damage by demyelation, results in some symptoms of numbness of skin, muscle paralysis, dilated nerves, and eye problems that could lead to blindness. And that's just because the facial nerves are affected and it can damage your eye cells. And then as for respiratory, you can get stuffy and bleeding nose. But there's an interesting thing that pain often isn't felt with this disease because of the nerve damage. There are some extreme nerve damage symptoms, not exactly loss of finger and toes as we see down here, more like a shrinkage of your fingers and toes, and that's because someone with this disease that was untreated, if they like run into a wall or if they hit their hand on a door, they're not going to notice that pain. So over time, their fingers can actually like shrink and shorten, which is kind of interesting. And then our risk groups, uh, Mycobacterium leprosy, it's super rare these days. So more than 95% of people, they actually have a natural immunity. And then approximately 2 million people globally are permanently disabled from the disease. And that's mostly because of neglect of treatment in developing countries. And then the most cases reported are in these developing countries like Democratic Republic of Congo, Nigeria, some places here in South America. Anything in the darker blue we see. And then treatments and preventatives. In the 1940s, there was a breakthrough with this antibiotic dapsone, but eventually leprosy built up a resistance to this drug. So some primary treatments that we use right now are rifampicin and clofazimine. Multi-drug therapies, basically they're taken together, and it's about a 6 to 12 months of treatment, so a bit longer, but you're no longer contagious after 1 week of multi-drug therapies. And then right now, there is no effective vaccine that's been approved, but our best preventative is to cover your coughs and sneezes, as shown by this little chart over here. That's pretty self-explanatory how to cover your coughs and your sneezes. And then here are my references. So that's going to conclude my presentation.